Right. Okay, can somebody confirm you can hear me?
Yes, we can hear you.
Thank you. Let me turn my volume up. Okay, before we begin, are there any questions? If there's no questions, we're going to be discussing Lab 7 today, throat and urine cultures. Am I sharing my screen?
No.
All right, that's actually what I want to do. So I want to briefly talk about the grading. I've done some grading. Let me get into the grade book, and that'll help me discuss. I got some extra credit study group graded. Oh, I need to mention I got the extra credit quiz graded. I think that's quiz 00A and 00B. 00A actually I had graded before, but you have to click on your grade to see the score. I'm not sure what's wrong with this. I've had a problem with it in the past, and it's not showing your score. It's showing like a document, like you submitted something and it's not graded. But if you click on it, you'll see that there is a score in there. And then the practice quiz 00B is graded. 0.1 point for each correct question. If you took the exam, 0 if you didn't take the quiz. Let's see, what else did I get graded? I've started grading lab 5, I think it is, but I'm only about less than halfway, maybe a third of the way through the class. I'm trying to get lab 5 finished, and I hope to get finished tonight. I'm not sure I'm going to succeed. That means lab 6 is not going to be graded, and you're supposed to have a quiz covering lab 6. So I think I sent an announcement out. Let me check that. Stating that I'm postponing the quiz 3, and that means I'm going to have to postpone quiz 4 because that's in a week later. And I'm going to give quiz 3 next Wednesday, meaning next week, instead of this week. And the reason is because lab 6 is on this quiz, and I don't have the first grading of it done yet. And I'm not going to because I'm still working on lab 5. So any questions about that? Well, the quizzes are going to be delayed by 1 week. I thought about trying to put out the answers, but then there won't be any point to grading the second grading of lab 6. So I decided not to do that. Let me see what else I have to say. I got the lecture worksheet done for, which one is it? Chapter 4. I have not graded worksheet lecture 5 yet. I hope to get that done tonight as well. All right, any question on the gradings? If not, I'll get out of the grade book and then we'll share my screen. I need to upload the new schedule. I haven't done that yet, but I've got it correct, I think. Let me check on that. I think it's correct in the Canvas webpage. Scrolling down, I guess I can share my screen now. Come on. Yeah, quiz 3C has been removed out of lecture week 6 now, and it's now in lecture 7. And then quiz 4 is on to lecture week 8. Like I said, I will upload the syllabus, hopefully sometime, maybe after the lab. All right, if there's no questions, let's move on to what we're doing today, Lab 7, throat and urine cultures. And hopefully I will send out your unknown number and your unknown results for the 3 tests sometime late tonight. It'll have to be after the 7 p.m. lab. And Monday, Wednesday, people, I sent it very late. It was actually after midnight, but just a little bit after midnight. So hopefully I'll get it out sometime tonight. I'm hoping to figure out how to email only the lab people. Actually, I can go into the lab and do it, I think. No, I've combined your labs, so I'm not sure how to send it to the 3 p.m. and the 7 p.m., how to separate it, but hopefully I'll figure that out and send it to just your lab. Otherwise, I'll have to send 2 files. One will be for the 3 p.m. lab and the other for the 7 p.m. lab. And I won't resort in that unless I can't figure out how to split it for the class. If you don't know, I've got the lecture and lab all together. So you guys are together in my Canvas website. So that's why I'm hoping to figure out how to send it only to the lab class. Any questions about any of that? All right, if not, let's go on to lab 7, throat and urine cultures and unknown results. I guess I can leave that up for the lecture. Lab 7, throat and urine cultures. There is some reading to do, as there is in every lab before the lab. Not too much for this lab, only page 392, and then the oxidase test on 132 and the catalase test on 155. The coagulase production, you can see the text at the end of this lab module. This lab module has a appendix, and you should read the appendix. There is some video clips in this lab. Oh, too far. Let me get up here. The learning objectives in this lab, first, be able to explain how to properly collect throat and urine specimens for routine laboratory culture. 2, list the common bacterial pathogens isolated from the throat and the urine cultures. 3, know the culture media used to cultivate these pathogens. 4, recognize which tests are used to identify throat pathogens and which tests are used to identify urine pathogens. 5, understand the principles and how they interpret to the following tests. We'll talk about, and you need to know the catalase test, the coagulase test, and the oxidase test. We will discuss the XV factor testing, but I'm not going to hold you to knowing it and understanding it, and you will not be tested over it. Any question about any of that? All right. Once specimens are received in the laboratory, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for throat and urine specimens. Below is a table overviewing the common media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For throat, the main media used is blood agar, and this can grow the most common pathogen isolated from the throat, Streptococcus pyogenes, which does cause strep throat, by the way. We do grow the bacteria also on chocolate agar to test for Haemophilus influenzae, which you can find in the throats of young children and infants. Any question about any of that? Let me state, if you don't recall, chocolate agar is very similar to blood agar. It is the same media and you're adding the same blood. It's just that when you're making blood agar, you allow the agar to cool to between 55 and 50 degrees. I think that's Celsius, but it could be Fahrenheit. I'm not sure about that. I should look that up. Anyways, you allow the agar to cool and then you add the blood and then the blood is not lysed. When you make chocolate agar, you take the agar directly out of the autoclave. And when the agar is still steaming hot, so close to 100 degrees Celsius, you add the blood, and that causes the blood to lyse, and the nutrients are released from the blood into the agar, and it makes the agar brown in color, why it's called chocolate agar. Any question about any of that? Chocolate agar can grow Haemophilus influenzae. Blood agar cannot. Haemophilus influenzae does not lyse the blood in blood agar, and it needs the contents of the blood to grow. Streptococcus pyogenes can lyse the red blood cells, and that's why it can grow in blood agar. All right, for the urine pathogens, these are pathogens which you have from either the urethra or the bladder or the ureters or as far up as the kidneys. And if you get a kidney infection, that's actually a very serious disease. The bladder is not as a serious a disease, but you don't want it. And obviously, if you have a pathogen in your urethra, you don't want that either. We use at least 2 media to grow the urine cultures. You always use blood agar, and that can grow both gram positive and gram negative, and it grows the fastidious pathogens as well as the non-fastidious. And then we also plate the urine culture on either McConkey's agar or EMB. So you always plate it on blood agar, and then you always plate it on one of the other, McConkey's agar or EMB. McConkey's agar and EMB grows only gram negatives, and that is what the majority of the urine pathogens are. They're mostly from the family Enterobacteriaceae. The most common pathogen in the urine cultures is actually E. coli. But you can get other pathogens which are gram positive, such as Streptococcus, Enterococcus, and Staphylococcus. And so we do need to grow it on blood agar. You can get other gram negatives. They're mainly Pseudomonaceae, the Pseudomonas, and we'll talk about distinguishing the 2 gram negatives from each other. Any question about any of that? All right, let's talk about throat cultures. Sore throats can be due to either viruses or bacteria. In this lab, we're only going to be talking about bacterial infections causing sore throats. The most common bacterial pathogen from a sore throat is Streptococcus pyogenes. It's also given the name Group A strep or gas for the abbreviation Group A strep. I don't use that terminology. Group A Streptococcus is looking at the antigens of Streptococcus, and it's not looking at the species name. I use the species name, and the two are the same. It's just that I just don't use the terminology Group A strep. You may use that terminology if you like. I would rather you didn't use gas, but you can use that terminology too. When you look at a bacterial sore throat, if it's red, the likely infective agent is a viral, swollen tonsils, red throat. If, on the other hand, it is red with white patches and white spots, that is likely a bacterial infection. And you can particularly see the white spots on the tonsils and the back of the throat, but you can see whitish or gray spots on the tongue as well. And you will see redness in the sore throat. That's a bacterial infection as well. Strep throat is a problem because the patient oftentimes cannot throw strep throat off. Strep throat, the bacteria, Streptococcus pyogenes, puts out a capsule, and that protects the bacteria from phagocytes. It can protect the bacteria from other defenses, like antibodies, for example, don't bind to Streptococcus pyogenes as well with the capsule. But especially phagocytes, the white blood cells, they're not able to engulf the bacteria. And that's why strep throat is very hard for a patient to throw off. To test the throat for a bacterial infection, you want to use a tongue suppressor to hold the tongue out of the way, and then streak the back of the throat and the tonsils. You do not want to streak and get a culture of the sides of the mouth, the tongue, or the roof of the mouth. Patients that test positive for Streptococcus pyogenes are put on antibiotic therapy because, like I stated, most of the time these patients cannot throw off the strep throat. Streptococcus pyogenes is a fastidious organism, meaning it must be cultured on blood agar, and that's with TSA with 5% sheep's blood added. The plate is then grown, and then you examine it for small white colonies that have clear zones of hemolysis, meaning beta hemolysis around the colonies. If you do see that, that's likely to be Streptococcus pyogenes, but it could be some other beta hemolytic bacteria. And there is another member of the family Streptococcus which causes beta hemolysis. Only 2 biochemical tests are required for definitive identification of Streptococcus pyogenes. Once you have beta hemolysis on blood agar from a strep throat. So this would be a throat culture that is not showing beta hemolysis. That's actually alpha hemolysis because of the darkening right there. And I don't really see it on the individual colonies. So that's probably a weak alpha hemolytic bacteria, where you're seeing alpha hemolysis, but only where the bacteria are very heavy. And we're not really seeing it right in this region here. There's a little bit around that one, but it's pretty weak. And this is beta hemolysis, where you can see right there and right there, that white little tiny colony is the bacteria. And then there's a large ring of beta hemolysis or complete hemolysis around that colony. And there you can see complete hemolysis right there and there and there and there. Any question about any of that? All right, so that's likely to be Streptococcus pyogenes. Only 2 tests are needed to show that that is Streptococcus pyogenes. First, you run the catalase test. And catalase takes hydrogen peroxide in the presence of the enzyme to make water and oxygen. The oxygen creates bubbling in the hydrogen peroxide. So if you see bubbling right here, that's positive for the catalase test. And if there's no bubbling, then that's likely to be Streptococcus or Enterococcus that is no bubbling. Most bacteria that grow in the air, meaning grow on a petri dish, produce catalase. However, Streptococcus and Enterococcus can grow in the air, and they are lacking catalase. Any question about that? So those two families lack catalase, they'll be catalase negative. Most other bacteria that grow in the air are catalase positive, and you'll see bubbling like that, at least on the slide test. You then take the culture, that is beta hemolytic, and spread it over this region of the plate. And then you put the bacterial, no, the bacteria, the disc containing the antibiotic bacitracin on. You put it on 5 minutes after streaking the bacteria on, and then incubate it in the incubator and look at the growth pattern. If you have a zone of inhibition around the bacitracin disc, then that is Streptococcus pyogenes. It is sensitive to the antibiotic bacitracin. If, on the other hand, the bacteria is resistant to the bacitracin, either there's no zone of inhibition or the zone of inhibition is below a certain size, it is resistant to the bacitracin, and that is not Streptococcus pyogenes. Any questions about how we prove that a beta hemolytic strep bacteria is Streptococcus pyogenes? All right, another organism that can be clinically significant, isolated from the throat of small children or infants, is Haemophilus influenzae. Haemophilus influenzae is a gram-negative coccobacillus. You can read more about Haemophilus influenzae in the appendix of this lab module. Haemophilus influenzae is one of the most common causes of ear, eye, and sinus infections in babies and young children. Haemophilus influenzae is a fastidious organism, gram-negative coccobacillus, that requires the growth factors, the V factor, which is actually NAD, we talked about that coenzyme, and Hemin, the X factor, which are supplied in chocolate agar, but not in blood agar, because Haemophilus influenzae cannot release the agents from the blood. They do not lyse the red blood cells. So we can grow Haemophilus influenzae on chocolate agar. We cannot grow it on blood agar unless we streak Staphylococcus aureus on the blood agar in a streak like here, and then you streak the Haemophilus influenzae this way on the blood agar. And as you can notice on the streak, let me blow this up. In the streak pattern over here, we see no Haemophilus influenzae growing. Only around the Staphylococcus aureus do we have Haemophilus influenzae growing. And what's happening is the Staphylococcus aureus is lysing the blood, and then the reagents needed from the blood are diffusing through the agar, and they're diffusing to this region of the agar. And so you can grow Haemophilus influenzae if you have Staphylococcus aureus growing nearby on the blood agar, but only around the Staphylococcus aureus can you see Haemophilus influenzae growing. Any question about any of that? All right. Let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infections, cystitis, a bladder infection, urethritis, urethra infection or polynephritis, and that would be either the ureters infected or the kidney infected. Normally, the bladder, they used to say, is sterile, but as you know, the bladder of females is not sterile. There is normal microbiota growing in the bladder of females, and I looked it up to see if there is a normal microbiota in the bladder in males, and nobody studied it, so I don't know the answer. Okay, and they've known for about 10 years that females have normal microbiota in the urine, in the bladder. But like I said, in the last 10 years, nobody studied it in males, so I don't know the answer. Is there a normal microbiota in the males? And I just don't know. Okay. Females are more predisposed to a urinary tract infection because they have a shorter urethra than males. To sample and obtain a urine culture, you first clean the genital area with usually alcohol or some other non-alcoholic sanitizer, and then start voiding the urine, and you catch the urine midstream. This is called clean catch midstream, where you only collect the middle portion of the urine stream, and that's to reduce the bacteria that come from the urethra. The urethra is loaded with bacteria, and you will get it in your urine sample if you void it, but if you use the clean catch midstream, you'll reduce the number. You can get a cleaner sample by inserting a catheter into the bladder and then sampling the urine directly. However, the urethra has bacteria on it, and then when you put the catheter up the urethra and into the bladder, you can put bacteria from the urethra into the bladder and then sample that. So the catheter isn't perfect. You will get contamination from the urethra with the catheter too. The only way to sample the urine directly without getting any contamination is a suprapubic aspiration, where you put a needle through the abdominal wall into the bladder and then sample the bladder directly. Now, patients do not like that, and they don't really like the catheter too. They would much prefer the clean catch midstream method. Once you get a urine sample, let me make sure I've talked about everything here. I have, yeah, okay. You then place 0.001 milliliters of urine onto a blood agar plate. That is 1 microliter of urine. And we actually have a tool for measuring that. Right there is a urine culture tool with a loop that holds 0.001 milliliter, which is 1 microliter. And then you take that and you streak the blood agar this way to get growth on the blood agar. And then you take the urine specimen and you grow it on either McConkey's or EMB as well, to only get the gram negatives to grow on the EMB or the McConkey's. Any question about any of that? We put it on McConkey's or EMB to grow specifically coliforms, which are the most numerous specimen in the urine cultures. And the number one most common cause of urinary tract infections is E. coli, which is a coliform. And these are all the family Enterobacteriaceae. But there are other specimens that do come out of the urine cultures, and there are gram positives, so we do need to grow it on blood agar, always. And then either McConkey's or EMB to remove the gram positives and look at the gram negatives. For a positive urine culture, the result is multifaceted. Numerous factors have to be considered, and that is the type of collection. Was it a clean catch voided midstream, or was it suprapubic aspiration or catheterization? Because all of the methods, except for the suprapubic aspiration, will have contaminating bacteria from the urethra. You look at whether the patient is male or female. Females have more contamination in the urinary sample than males, if you have like clean catch midstream. And then you look at the number of colonies per mil of urine, and you look at whether there's polymicrobial growth. If you see lots of multiple organisms isolated, it may indicate that the collection technique was poor. Any question about any of that? All right, once you get your samples growing on the blood auger and or on the McConkey's or EMB auger, you then run some tests to identify the species. The first test we're going to talk about is the coagulase enzyme. This enzyme is used to differentiate Staphylococcus aureus from other members of the Staphylococcus species. Staphylococcus aureus is coagulase positive. Other members of Staphylococcus, meaning the other members of the family, are coagulase negative. Coagulase contributes to the pathogenicity of Staphylococcus aureus. Coagulase causes a blood clot to occur by taking the fibrinogen in the presence of coagulase, making the fibrin clots, which will then cause the blood to clot. This can protect the Staphylococcus aureus when the bacteria infection is first getting started and is low in numbers. The immune system has to eat through the clot before they can attack the Staphylococcus aureus. So the blood clot is actually a way the Staphylococcus aureus protects itself when it's first causing an infection. To test the coagulase test, there are two tests you can use, the slide test and the tube test. In the tube test, if you have clumping of the serum, then the bacteria are coagulase positive. If the serum does not clump, stays a liquid, as in this case, it's coagulase negative. On the slide test, the plasma stays liquid and, I don't know, smooth, non-clumping. And if it's coagulase positive, you get clumping on the slide test. All right, as I've mentioned, the most common bacteria pathogen isolated from a urine culture is E. coli. E. coli is of the family Enterobacteriaceae. We can distinguish the Enterobacteriaceae from other gram-negative bacilli that are causing the urinary tract infection by the oxidase test. The members of the Enterobacteriaceae family, including E. coli, are oxidase negative. The members of the Pseudomonaceae family, particularly Pseudomonas aeruginosa, is oxidase positive. And so we can use this test to separate out the Pseudomonaceae family from the Enterobacteriaceae family. Both of these families are gram-negative bacillus that cause UTIs. The families do differ besides in the oxidase test. The Pseudomonaceae family, they're all aerobic respirers, meaning they have to grow in oxygen. The Enterobacteriaceae family, they're all facultative anaerobes, meaning they prefer to grow with oxygen and will engage in aerobic respiration if oxygen is present. But if oxygen isn't present, they'll fall back on anaerobic respiration. The Pseudomonaceae family are all non-glucose fermenting organisms, and the family Enterobacteriaceae all ferment glucose. So there's actually 3 tests you could use to separate the two, but the oxidase test is the one that we normally use. For one thing, it's very quick. You just grab the bacteria and then put the bacteria on the oxidase reagents. And then a short time later, you look at the test. Let me show you that. And if it's purple, that's oxidase positive, or blue color, it is oxidase positive. In this region right here, it is oxidase negative, and there is no purple or blue color. And like I said, the Pseudomonaceae are all oxidase positive. The Enterobacteriaceae are all oxidase negative. Oxidase is looking for the presence of cytochrome C, this enzyme, or what do you call that? Enzyme cofactor, coenzyme, there we go. Enzyme coenzyme is present in the electron transport chain of aerobic respiration. And if that enzyme is present, we will get the oxidase test being positive. Any question about any of that? Let me see. No, that's it. All right. All right. In summary, urine and throat cultures are 2 of the more common types of specimens received in the lab for bacterial culture, and microbiologists are trained to isolate and identify the pathogens present. You can skip the references, but you should know that this lab, you're not going to be performing any of these activities. However, you will be held responsible for knowing how to perform the activities in this lab. We have a little video for you to watch on running the catalase test, another little video on running the video on the coagulase test, and a little video on running the oxidase test. The interactive virtual labs are not working. They require flash, and that doesn't work anymore. All right, any questions about any of that? If not, let's go on to the worksheet. This lab only has a few questions, 13, although there's like 5 questions to 13. There are no case studies in this lab, so this is a easier and quicker lab to do. Question 1, name the enzyme, catalase, coagulase, or oxidase, that converts fibrinogen to fibrin. That's 1A. And then 1B, name an organism that tests positive for this enzyme. 2A, name the enzyme that detects the presence of cytochrome C proteins in the electron transport system. Name 2 organisms that test positive for this enzyme. And it's mentioned in the lab module as well as in the video clip. In 2B, name the 2 organisms that test positive for the enzyme. 3A, name the enzyme that hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen. And then name 2 organisms, this is 3B, that test negative for this enzyme. And in this case, you can mention 2 species or 2 families. Normally, you have to mention 2 species, but in this case, you can mention 2 families. 4, still looking at the 3 enzymes that we mentioned above. Which rapid test, meaning which enzyme, is used to differentiate between Staphylococcus and Streptococcus? Which rapid test or enzyme is used to differentiate between Staphylococcus aureus from other members of the Staphylococcus family? And then 3, which rapid test or enzyme is used to differentiate between Enterobacteriaceae, such as E. coli, and the Pseudomonaceae, such as Pseudomonas aeruginosa. 5, concerning specimen collection for routine throat culture, what areas of the throat should be swabbed, 5A, and 5B, what areas of the mouth should be avoided? 6, what is the most common bacterial cause of pharyngitis? That's a sore throat. And you do need to say a bacterial cause. You cannot say a viral cause. 7, what test is used by microbiologists to differentiate between beta hemolytic group A streptococcus, meaning Streptococcus pyogenes, from other beta hemolytic streptococcal species isolated from throat cultures? And because we're only separating out Streptococcus, meaning Streptococcus pyogenes from other members of the family Streptococcus, you don't need to use the catalase test because both of them would be catalase negative. All right, 8A, state the 2 non-selective agar plates that are inoculated concerning a child's throat culture. And we're talking about a small child here. 8B, what organism can be isolated on CAP, that's chocolate agar, but not on BAP, and that's blood agar? And 8C I've crossed out because it's not in the lab module. You'd have to look it up online, so you do not need to answer 8C, but you can if you want to. 9, you don't get any extra credit, but you can answer 9C if you want to, but you don't need to. 9, list 3 methods that are used to collect urine for routine culture. 10, list 3 reasons why urine specimens should be rejected for culture. And that is discussed in the lab module. But if you don't find it, you can just look online. It is in the lab module, though. I just looked in the last lab. That'd be Wednesday's lab, Monday's lab, whatever. What is today? Tuesday. So I looked in Monday's lab. 11, how much urine is placed on the surface of a blood agar plate? And 12, what is the most common bacteria isolated from patients with cystitis? That is a urinary tract infection. 13 says identify the Gram reaction, that's the Gram stain, and the cell morphology, that is the shape of the bacteria, not the arrangement of the bacteria, of these species. So 13A, what's the Gram stain and the morphology of Staphylococcus cephrophyticus? You can find this in the lab module if you need to, or in the appendix. What is the Gram stain of E. coli and the cell morphology? And that one you should actually know from a previous lab. What is the Gram stain and the cell morphology of Pseudomonas aeruginosa? That is discussed in the lab module. What is the Gram reaction of Streptococcus pyogenes, meaning the Gram stain and the cell morphology? That's discussed in the lab module. And what's the Gram reaction and the cell morphology of Haemophilus influenzae? This is in the lab module as well as in the appendix for this lab module. Let me briefly just show you the appendix, and then I'll turn it over to you guys to work in the lab. So the appendix, we have a section talking about Streptococcus. Come on. My mouse isn't working. And beta hemolytic streptococci, especially Streptococcus pyogenes. And then we have a little section talking about the Haemophilus family, specifically Haemophilus influenzae. And I can state that this is actually misnamed. It was named by Louis Pasteur, who thought that this bacteria was responsible for causing the flu, but he never used Cole's postulates to prove it. And what he did was he sampled people who had the flu and he obtained Haemophilus influenzae from them, but Haemophilus influenzae does not cause the flu. We have viruses that cause the flu, by the way. And then there's a little section talking about the coagulase test because the lab module was pretty slight on talking about it. All right, any question about this lab? If not, I'll be here until let's make it 4:15, 4:20 to help you with the lab. If you're a student that gets a lot of questions wrong in the lab, you should take advantage of my ability to help you in the lab. So if you have a question on this lab or any other questions, please ask during the lab. I will be eating, but you may certainly ask a question and I will be here until the last student logs out, or let's say about 4:20. Any questions? All right, get started on the lab.